My dear students of 11th class, and previously we have talked about the photosynthesis and what we have been taught about the photosynthesis in our previous classes. And today we will move ahead with the different types of the experiments that were performed by different scientists at different times in the past so as to reveal what actually the photosynthesis is, what are the various types of raw materials that are required for this particular reaction, photochemical reaction, and what are the factors or conditions that are required for the process of photosynthesis that is to be carried out inside the green cells of the plants. So it was Priestley whose series of experiments in 1770 revealed the presence of oxygen in the air, and in fact this Priestley is considered to be the discoverer of oxygen gas. Priestley revealed that the essential role of the air in the growth of the green plants. He observed that a candle burning in a closed space or a bell jar gets extinguished. Similarly, a mouse would soon suffocate in a closed space. And then he concluded that the burning candle or an animal that breathed the air both somehow damaged the air. But when he placed a mint plant in the same bell jar, he found that the mouse stayed alive and the candle continued to burn. Priestley hypothesized as follows: plants restore to the air whatever breathing animals and burning candles remove. And using a similar setup as one used by Priestley, but by placing it once in the dark and once in the sunlight, Jan Ingenhousz, born in 1773 and died in 1799, showed that the sunlight is essential to the plant process that somehow purifies the air polluted by burning candles or breathing animals. Ingenhousz is in an elegant experiment with an aquatic plant showed that in bright sunlight some bubbles were formed around the green plants, while in the dark they did not. Later he identified these bubbles to be of oxygen. Hence he showed that it is only the green part of the plants that could release oxygen. And it was not until about 1854 that Julius Van Sachs provided evidence for production of glucose when plants grow. Glucose is usually stored as starch. His later study showed that the green substances in the plants, that is the chlorophyll as we know it now, is located in special bodies later called chloroplasts. You must have heard about the chloroplasts within the plant cells. He found that the green parts in the plants where glucose is made, and that the glucose is usually stored as starch. And now consider the interesting experiment that was done by T.W. Engelmann. He was born in 1843 and died 1909. Using a prism, he split light into spectral components and then illuminated a green alga, cladophora, placed in a suspension of aerobic bacteria. The bacteria were used to detect the sites of oxygen evolution. And he observed that the bacteria accumulated mainly in the region of blue and red of the split spectrum. A first action spectrum of the photosynthesis was thus described. It resembles roughly the absorption spectra of the chlorophyll A and chlorophyll B that is to be discussed in the forthcoming lectures. And by the middle of the 19th century, the key features of plant photosynthesis were known. Namely, the plants could use light energy to make carbohydrates from carbon dioxide and water. The empirical equation representing the total process of photosynthesis for oxygen evolution organisms or the oxygen evolving organisms, it was understood as carbon dioxide plus water in the presence of sunlight gives H2O plus oxygen, where CH2O represents a carbohydrate that was later found to be glucose, a 6-carbon sugar. And then a milestone contribution to the understanding of the photosynthesis was that made by a microbiologist, Cornelius van Niel, born in 1897 and died 1985, who based on the study of the purple and green bacteria demonstrated that photosynthesis is essentially a light-dependent reaction in which hydrogen from a suitable oxidized compound reduces carbon dioxide to carbohydrate. Like this, 2 hydrogen from a source plus carbon dioxide gives 2A, that is released from the compound of hydrogen and that particular substance, and then CH2OH and carbon and water. In green plants, water is the hydrogen donor and is oxidized to oxygen. And some organisms do not release oxygen during photosynthesis. When hydrogen sulfide instead is the hydrogen donor for purple and green sulfur bacteria, the oxidation product is sulfur or sulfate, depending on the organisms and not on oxygen. Hence, he inferred that the oxygen evolved by the green plants comes from water, not from carbon dioxide. And this was later proved by using radioisotopic techniques. And the correct equation that would represent the overall process of photosynthesis is 6 times carbon dioxide plus 12 times water in the presence of light. It gives glucose, that is C6H12O6 plus water plus oxygen. Thanks. Have a nice day.